Right, so I know that maybe for weather some people still are coming, but I think we have to get started because actually I'm gonna start.
Yes.
Yeah, so otherwise we cannot finish everything. So all right, good morning everyone. So how is everyone doing today? Good? Tired? Exhausted? Okay, so today we are going to actually cover a very exciting topic. Okay, microbial electrochemical systems for waste and wastewater treatment. So far over last 3 days we kind of focused on more on conventional bioprocesses for waste and wastewater treatment, and today we are going to see a very emerging process. Okay, microbial electrochemical technologies. We are going to see like how we can actually use this process for waste and wastewater treatment. So anyone here familiar with microbial electrochemical systems before? No? So microbial electrochemical systems, actually that is an environmental bioprocess. Okay, so in conventional environmental bioprocess, what we do, we utilize microbial metabolism, right? So here, in addition to microbial metabolism, actually we add electrochemistry there. Okay, so we combine microbial metabolism with electrochemistry. So basically, when we talk about electrochemistry, it's electrochemical cell, right? That we can add. So basically, like an electrochemical cell with anode and cathode, and you can have oxidation reduction reaction. So for example, you can have on anode you can have oxidation, on the cathode you can have reduction reaction. And but one of the unique feature of microbial electrochemical system is here we use a unique type of bacteria. Okay, so those bacteria, they are electrochemically active. We know that actually like all microbes, they transfer electron, right? Like we learned, they actually kind of transfer electron from electron donor to electron acceptor, right? So there is electron transfer there, but this unique type of microbe, electroactive microbe, their respiration process in a sense that, okay, they can, so like for example activated sludge process, you provided oxygen as electron acceptor, right? But here they can actually transfer the electron to electrode surface. Okay, so that means some insoluble conductive or solid material, okay, they can use as electron acceptor. Okay, so that's the unique feature of microbial electrochemical technologies. But actually there are many terms actually over last 20 or 25 years researchers have used many different terms. Okay, and sometimes actually it's very confusing for someone actually start working in this area for first time. So for example, like there are many different application of microbial electrochemical systems. So for example, microbial fuel cell. Microbial fuel cell, you can actually produce electricity like like a regular electrochemical or regular fuel cell. You can actually produce microbial fuel cell can produce electricity using electroactive bacteria. So we call it MFC. We can actually have we can apply some voltage, okay, and we can get a wide variety of product like including biohydrogen from microbial electrochemical system. We call it microbial electrolysis cell, MAC. Okay, MEC. Then some researcher found that, okay, we can actually apply this concept for desalination of water. Now people call this microbial desalination cell, MDC. Okay, then researcher found actually we can actually produce different types of value-added product on the cathode side, okay, using carbon dioxide as carbon source, and that process is called microbial electrosynthesis, MES. So now lot of new application. Okay, so that's why then researcher thought, okay, we kind of give a common name. Okay, we call it MXC. So that X may represent different application. It could be fuel cell, it could be electrolysis cell, it could be desalination cell. Like then actually researchers, they have shown that you can do denitrification using this concept. So now MDC for microbial desalination cell, MDC for microbial denitrification cell, right? So there are a lot of overlap, many names, then we kind of end up with using MXC, okay? And any technology that is based on this concept, we call it MAT, microbial electrochemical technologies. Sometimes we also use the term called bioelectrochemical cell or bioelectrochemical systems, okay? So how that works, okay, like any traditional environmental bioprocess, we have oxidation reduction reaction, okay, but here that thing actually happen on the electrode, okay. So what happened, like when actually we provide those microbes EAB, so we have anode and cathode electrode and two chamber. Sometimes depending on the application, anode and cathode chamber can be separated by membrane, and typically those electroactive bacteria, they produce actually biofilm, they kind of form biofilm on the anode surface. So when you actually provide them with electron donor, they can actually oxidize those electron donor and transfer that electron to the anode surface, okay, anode surface, and the process actually also produce carbon dioxide and proton, and then it kind of transfer electron to the anode surface. Then actually because of potential gradient, that electron can transfer from the anode to the cathode, okay. And cathode, you can manipulate the terminal electron acceptor, right? So like for example, like activated sludge process, you provided oxygen and oxygen was reduced to water, right? So depending on the application, we can manipulate like what we want from the microbial electrochemical system. We can manipulate the terminal electron acceptor here and can get different types of value added product, okay. So when actually electron transfer from anode to cathode, to maintain charge neutrality, the proton that is actually released from the oxidation of your electron donor, proton kind of also transfer from anode to the cathode, because one negative charge is going there, so one positive charge should also go there to maintain the neutrality, right? So and going back, that electroactive bacteria, okay, they also actually researchers, they also gave them different name, okay. So some people call them electroactive bacteria, EAB. Some researchers actually name them anode respiring bacteria, because they can do respiration on the anode surface, okay. So they give them a name in anode respiring bacteria, ARB. Some researchers call them exoelectrogen, okay. So why exoelectrogen? Like like you remember the name from anaerobic digestion, methanogen, right? So microbiologists, they kind of like to actually name microbes, okay, like some sort of like gen, right? So those microbes, they can do extracellular electron transfer, so that's why researchers name them exoelectrogen. So again, there are many different names. So couple of years ago, the research community, they agreed that okay, what, like it's better to call them electroactive bacteria, because now we know that actually they can be not only just anode, they can be in other engineered system as well, okay. Sometimes even they can be on the cathode as well. So maybe it's better to call them like electroactive bacteria or EAB, okay. So even when I back in like between 2012 and 2015, when I actually did my PhD, I also used to use the term like anode respiring bacteria at that time, but now we call it EAB, electroactive bacteria. So here on the cathode, you can manipulate the terminal electron acceptor and you can get different type of product. So for example, like in so there are actually oxidation on anode, and cathode you have reduction. So for example, if we provide acetate as substrate, okay, acetate as substrate for or electron donor for microbes, you can see this half reaction. Like in very first lecture, we learned about half reaction, right? So if acetate is oxidized, it's gonna release carbon dioxide, proton and electron, you can see here, right? So this is oxidation. So in cathode, if I just provide oxygen as electron acceptor, then oxygen gonna be reduced to water. Okay. So we can actually, so this is kind of like the basic reaction, but we can have like different electron acceptor on the cathode side, and we can have different product that we want. So this kind of electroactive bacteria was actually first found in natural systems. So one researcher actually, he is a microbiologist, okay, in US. So his research team actually found this kind of microbes in sediment, river sediment in US, one river in US, and they found these kind of microbes, okay, called geobacter species, have some unique feature. So they can actually oxidize ferric to ferrous iron, okay, using extracellular electron transfer. Okay, so they can actually convert ferric to ferrous iron, and at that time actually people didn't know that actually they can also work as a electroactive bacteria and microbial fuel cell. But when microbial fuel cell research became very popular and people start to actually do microbial analysis and try to know, okay, what kind of microbes are there, they found that actually, in most cases geobacter species is a common type of microbe. Most of the time we find on anode surface of microbial electrochemical cell. So EAB, they can respire, so they can do respiration. So this is actually a unique example of anaerobic respiration. Okay, so anaerobic respiration, and because they can use insoluble electron acceptor like metal, minerals, like in this example they used metal, right? But in our case, it's when we talk about microbial electrochemical cell, they are using electrode, right? They are actually transferring the electron to electrode. So, and EAB, they use unique electron transfer mechanism which is called extracellular electron transfer. Okay, so they have they oxidize the organic compound and transfer that electron to their outer membrane electron transport system, okay, like other microbes, but then that electron is actually transferred to the electrode surface, right? And that part is called extracellular electron transfer, or EET. Okay, so because of this feature, okay, they are kind of considered as unique. So in addition to geobacter, other common type is Schwannella species. They are another type of bacteria that can do is actually this kind of EET. But later on, there are many other bacteria have been discovered, okay, they can do EET, but we like maybe 10 or 15 years ago it was not known that actually those microbes can also do EET. Okay,
so these are, we can say, anaerobic bacteria?
Yeah, they are anaerobic bacteria, yes. So they don't need oxygen, right? So they, without oxygen they can do their respiration, so they are anaerobic bacteria. So here you can see here, there are three major steps involved. Okay, three major steps involved, electron transfer from electron donor to the electrode surface. So for example, first step, microbe they oxidize the electron donor, release electron, and then intracellular electron carrier NAD plus NADH. Okay, this is a redox cofactor or can act as electron carrier. So they transfer electron to the outer membrane protein, which is kind of microbial electron transport chain system, okay, or we call it ETS. And then from outer membrane protein, okay, electron is transferring from the outer membrane protein to the electron surface, which is called EET. Okay, EET. So that part is kind of like the unique feature of this kind of microbe, because sometimes what happen, a microbe, they can be very far from the electrode, okay, but they can still transfer electron to the electrode surface. Let's say you have what is the size of microbes? Few micrometer, okay, but you can have 100 micrometer biofilm. Okay, biofilm thickness could be 100 micrometer, but those microbes still can oxidize electron donor and transfer electron to the electrode surface, like long distance electron transfer. So that is called EET. Okay, so that's why it's so unique. So how microbes they do EET? Okay, so not a single mechanism. Microbes they can use many different mechanism, okay, or combination of multiple mechanism. So first researcher actually thought maybe like those microbes when they are actually form biofilm on the electrode surface, so there is some direct contact with the electrode and they can actually transfer electron. So for example, I would say like if we draw this diagram, so researcher first thought okay, microbes they are going to be in direct contact with the electrode and they can actually transfer electron. But now most of the time researcher found actually like monolayer. So this is kind of monolayer biofilm, okay, monolayer biofilm. You have a single layer of microbes. But researcher found okay, you know what, like when we have multi-layer biofilm, actually multi-layer biofilm can produce even more electricity. So then they thought okay, you know what, that means those microbes they are actually outer layer of the biofilm, they can do long distance electron transfer. So they are not necessarily those microbes need to be actually attached to the electrode surface, right? So then they decided that direct contact is not enough to produce high current density from this kind of system. So other mechanism might be active there. Then researcher found actually microbes they can use some soluble redox material, okay. So redox active material. So what is redox active material? They can be either oxidized or reduced, okay. So we call them actually redox mediator or shuttling compound. So how that works? Actually, so let's say you have some electrode, okay, and you have some microbes in suspension, okay. Now if you have this kind of redox mediator in your system, what's going to happen? So those are redox active compound. They can go to the outer membrane protein, okay, outer membrane protein of microbes, and they can be reduced. They can take the electron from the microbe and can go to the electrode surface and can be oxidized. So from oxidation they are going to release the electron and then they go back again and then reduced again, come to the electrode surface, then go back and forth, okay. But those are soluble compound. So think about if you have a continuous reactor, okay, continuously you are actually like feeding that with wastewater, those compound may be washed out from the system, okay. So some researcher they did experiment, they added this kind of material, okay, in the reactor, and then they found actually current can be increased in batch mode, but when they actually operate in continuous mode, okay, it's a kind of like don't show high current density. So then researcher realized okay, that mechanism may not actually lead to high current density from this kind of system. Then in actually I think it was in 2007 or 2008 researcher found actually, or maybe 2005, okay, almost 20 years ago, researcher found that actually those microbes can actually produce hair like appendages, protein appendages, okay. So they can actually produce some wire and make a connection with the electrode surface. So even they are outer layer of the biofilm, they can actually produce some wire, okay, and use that wire to actually transfer microbes, and those are actually like produced by microbes. So sometimes we call them nanowire produced by microbes, or some researcher actually call them pili, so semiconductive pili, and researcher actually confirmed that those hair like appendages actually they are semiconductive in nature. And then another actually mechanism have been proposed. So what is that mechanism? It's called electron hopping. So what is electron hopping? Electron is jumping from one hole to another hole, right? So what they actually proposed, that there could be, instead of this kind of soluble compound, okay, there could be some compound in the biofilm, they are also redox active and they are very close to each other. So what happened, they can be, you know, like oxidized and reduced. There could be a chain reaction of oxidation reduction, and ultimately electron can reach the electrode surface. Okay, I know that what I explained may be still not clear, so let's see some figure. So first mechanism, direct contact. So microbes gonna be in direct contact with the electrode surface, but we know that actually that cannot produce high current density. Redox mediator or shuttling compound can be another option, okay, but they are soluble, so they can be washed out from the system. They can be either oxidized or reduced. So on the outer membrane protein, they are going to be oxidized or reduced.
Outer membrane protein oxidized.
They are going to be reduced. They are going to take electron,
Yeah.
right? And then on the electrode surface, they are going to be oxidized. Okay. So and going back, they are going to be reduced again and oxidized. So they are redox active compound. Okay. So they can be either oxidized or reduced if needed.
Give some example of this.
It's kind of, I forgot the name, but researcher found that some microbes can produce this kind of compound. But I can get back to you. Maybe tomorrow I can give you some name. Okay.
Do you think NAD plus could be such compound?
No, it's NAD plus NADH actually. This is intracellular compound, not extracellular, right? So,
Can you confirm or know that is pH has some kind of relation with this kind of mediator?
pH, okay, these kind of microbes, they prefer neutral pH. So, you know, like at low pH, if some of this kind of compound even active, microbes not going to have any activity. Okay.
That could be another challenge in wastewater treatment.
Yeah, so pH has to be close to neutral pH. Okay. So those microbes are actually very sensitive about pH. But now you can see like the microbial nanowire, those microbes, even they are actually far from the electrode, they can produce those hair like appendages and transfer electron. And then another mechanism is when they are actually far from the electrode, they can also use some redox cofactor. Okay. So redox cofactor, so they are actually like mediator, but they are immobilized in the system, immobilized within the biofilm, and they are very close to each other. So electron can be actually, you can have a chain reaction of oxidation reduction. Although here you can see those cofactors are very far, right? So I have one cofactor here, one cofactor here, but in practically they can be within like nanometer distance. Okay, so nanometer distance, so they can actually exchange electron.
So how they use this pili for electron transfer? Because as much as I know, these are sex pili. The bacteria are using these pili for conjugation.
It's yes, but this is actually a little bit different. Those are not yeah, like the researcher found actually their I think structure is quite different, you know, like quite different, and they are not actually found in all my bacteria. Okay. So the mechanism C and D, so microbial nanowire and electron hopping, we consider this method as conduction based DET. So microbes, that biofilm kind of, it's kind of act like a semiconductive material. Okay. So some researchers also call it not semiconductor, they call it metal like conduction. Okay. But it's actually, it has been found that microbes, they can form a biofilm. Okay. And that is also kind of semiconductive in nature. So I'm not sure whether you can see here, but you can see, like, you know, let me see if I can zoom in. Do you see it now? You can see some lines, right? So those are actually pili. So this is microscopic image of pili. So microbes, this is actually produced by geovector. Okay. And then even Schwannella could also produce, you can see those Schwannella microbes, actually they also produce this kind of pili. Okay. And they are mostly protein. So then what researcher did, they separated, okay, they extracted those pili from the microbes, okay, and then what they did, they used actually atomic force microscopy and they applied some voltage, okay. So you know Ohm's law, if you apply some voltage, you should, there are gonna be some resistance, okay, and you should see current, okay. So if you, so you can see here they applied different voltage and then they found actually a very, like, you know, ideal ohmic response. So with increasing in voltage, current is increasing linearly. So this is actually like ohmic response. So that means it's kind of like, you know, those material, they are actually conductive. So this is kind of actually confirmed that microbes, they can produce conductive nanowire. Actually the same thing was done for Schwannella, okay. So I'm not sure whether you can see it. So they have 2 electrodes. So those are 2 electrodes, and then there is a like those nanowire that is actually extracted from Schwannella species. I'm not sure whether, let me zoom in. Now do you see a line? So this is actually a pili. So then they actually applied voltage. Then they applied voltage, okay, and then again they found actually almost similar pattern, okay. So with increase in voltage, your current is increasing, right? So that is actually confirmed that like geovector species, Schwannella species can also produce conductive nanowire. But the difference between geovector and Schwannella, actually researcher found that some Schwannella species, okay, they don't have actually so mutant deficient in gene, they don't have any gene for C-type cytochrome, okay. So they don't have any gene for C-type cytochrome. If they try with this kind of pili or nanowire from Schwannella species, you can see with increase in voltage and current line is flat. So for them actually, you know, like it's not showing any ohmic response. So that means it's not conductive. So then researcher actually found that actually for Schwannella species, C-type cytochrome would actually be the key factor in extracellular electron transfer, okay. So then researcher actually proposed the method ohmic, you know, like electron hopping. So what is electron hopping? So microbes, they are going to still produce nanowire, okay, but that nanowire contain lot of cytochrome C, okay, and they can actually kind of work as a like hole for electron, okay. So when they are, and as I said, like, you know, they're gonna be, although it look very distant from each other, but in reality they could be in 1 to 2 nanometer distance, and then actually electron from outer membrane protein can actually jump from one cytochrome C to another cytochrome C, okay. So although I'm saying it's jumping, like, you know, because it's, we are calling electron hopping, but in reality it's oxidation reduction reaction, okay.
So these cytochromes are produced by bacteria?
Yeah, they are cytochromes are produced by bacteria. So for geovector, so if I summarize, geovector, the whole nanowire itself, it's like an electrical wire, okay. But for Schwannella species, it's it look like electrical wire, but it's not. Mainly it's actually the work is done by cytochrome, okay. Then another researcher, he proposed another mechanism. So he said, okay, you know what, sometimes some microbes, they don't produce, okay, they don't produce actually nanowire, but still they can do long distance electron transfer. So that means they actually use some actually redox cofactor, they are immobilized within the biofilm, okay. So some when the microbes, they form biofilm, they can produce EPS, right. So they produce EPS, extracellular polymeric substances, that can actually help the biofilm formation. So some of those EPS material can act like redox cofactor as well, and that can actually also help you to transfer electron from outer membrane protein to the electron surface. So that is actually another mechanism of hopping. When microbes, they don't produce nanowire. Yeah.
Actually, you talk about the responsibility, and there is genes for C-type cytochrome, and they are producing this C-type, some protein from this C-type cytochrome, and I'm asking a question that so this C-type cytochrome is ubiquitous in plants too, but what are the difference between them and this type of microbes that they are showing conductivity, not any other plant? Why they are so specialized?
Okay, so if I understand your question correctly, okay, why this kind of microbes are unique?
Unique, because the cytochrome C is ubiquitous.
Okay.
So, what are the specialty that this type of microbes have?
That question I cannot answer. Why? Now I have to go back and see, who created bacteria and why they were actually provided with some unique feature. Okay, I can't really tell. Okay, but there might be actually, let's say, in natural environment, there might be some situation where those microbes, okay, they did not have, they were in some environment where they did not have enough electron acceptor. So they kind of, somehow to survive, okay, change their metabolic pathways. Okay. So that could be one of the reason, but honestly speaking, I cannot answer why those microbes have that.
Actually, I'm asking because I have some organism which shows this kind of property with another element. This is heavy metal. So this is a plant. So that is why it is also having this C type of cytochrome. But I don't know exactly what is the mechanism behind it. So when you say that there is this reason behind is the genes for C type cytochrome, I thought the same mechanism could be applied for my organism too.
So did you do any microbial analysis?
No, not
Okay, so maybe it's better to do some sequencing, okay, and you can see it's mixed culture, right, or pure culture.
just that.
So pure culture, you should know what kind of microbes.
No, it's actually plant, not microbe.
Okay. Honestly speaking, I cannot really answer that, but we can discuss that more, after the lecture, okay, if needed. So I'll move on. So microbes, they need some sort of conductive pathway for EET. So now then researcher thought, okay, you know what, now if conductive pathway is important, so the biofilm itself should be conductive as well. Okay. So now researcher tried if we can prove that actually biofilm is conductive. Okay. So how we can do that? Okay. So that was actually very smart approach. Okay. Published, I think this paper was published in about 2010 or something, this method was proposed. So you can have two electrode, okay, with some very small non-conductive gap. So they are actually two electrode on a glass base, okay, and there is a small non-conductive gap. So that non-conductive gap could be like 10 micrometer or 25 micrometer. So microbes, okay, so what is the size of microbes? Let's say 4 or 5 micrometer, right? So that non-conductive gap is about let's say 25 to 50 micrometer. But when microbes form biofilm, okay, so they can actually cover that non-conductive gap as well. Okay. So now, so here although it look like one electrode, but you have two electrodes. So what you can do using a source meter, okay, you can apply some very small voltage, and if that biofilm is conductive, then electron should be able to, because there is two electrode, they are not connected, right? The only connection they have is the biofilm. They are actually like connected by biofilm, not by any conductive material, right? So what I am saying here, you have two electrode. So let's say electrode A and electrode B, okay, and over time you have biofilm, they kind of grow. So and those A and B, they are not connected, right? There is no short circuit in there, and now they are actually, you have over time you have biofilm formation, okay, and you have the biofilm. When they form biofilm, you have BPS and other stuff that can actually cover that gap, okay. So now those two electrodes, they are only connected by biofilm. Now if you apply some voltage, okay, across A and B, if they are actually, the biofilm is conductive, you should see a ohmic response. Biofilm should actually act like an wire, okay, and then actually researcher confirmed that. They applied small voltage and they actually found like pili, they also found like, you know, very perfect ohmic response, okay. So voltage and current, okay. So now if you actually, that kind of confirm that if you see an ohmic response, that confirm that actually like biofilm is conductive. So if you know the biofilm thickness, and from you know like this response using Ohm's law, you can actually estimate conductance, and based on that, using an equation, you can actually you know like calculate conductivity, which is, let's say, millisiemens or microsiemens per meter, okay. And then the researcher group, actually it was, you can see all of them are published by one researcher, okay. So they tried different actually strain of geovector species, okay, and they found that actually for different geovector species, actually current density ampere per meter square actually show a perfect linear relationship with biofilm conductivity. So that means biofilm conductivity is very important to get high current density from microbial electrochemical system, okay. So conduction is the dominant pathway for high current density in microbial electrochemical system. So now before I jump into the next topic, so next topic, actually now we talked about fundamental, how that thing work, okay. Now we are going to talk about little bit of application, what are the different application, and then the third topic gonna be, majority of the portion gonna focus on microbial kinetics, and we are gonna learn, like we did some, we learned microbial kinetics on day 2 for activated sludge process, right? And I was telling you that if we learn the kinetics for one biological process, we can actually learn other biological process very easily, and you are going to see today actually how we are going to use like almost similar kinetic equation, and we are going to come up with an equation that can allow us to estimate current density from microbial fuel cell, okay. So but before I jump into this topic, any question? Yes.
This process can occur in all types of cells which have external electron acceptor, like you showed the example.
All types of external electron acceptor. Well, it depends, okay, what kind of process that you are looking into. Some of them might be thermodynamically possible, some of them may not be thermodynamically possible, okay.
I believe that electron acceptor is not the only necessity. It should be redox. Actually, it could handle redox reaction. Then only it can serve our purpose. Right?
So yes, sometimes, like, you know, I actually have some slide like explaining this, like, you know, thermodynamic significance there. So let's say you may consider, okay, you know what, I'm going to put that electron acceptor there, and this is the product that I want from this reaction. But the question is whether it's thermodynamically feasible or not. You have a redox potential for your donor, right? You have a redox potential for your reduction reaction, right? But now from this redox potential, you can tell whether it's thermodynamically possible or not. If not possible, that means you have to provide some energy from outside to overcome that thermodynamic barrier, okay?
Sir, but it should be possible for the survival of the cell.
Yeah, then yeah, if they don't have electron terminal electron acceptor, yes, microbes gonna actually
He's talking about the external electron acceptor, not internal. I guess you are explaining about internal electron acceptor.
so you are talking about external. Yeah, he is talking about external.
But you said about
I am also talking about external on cathode.
how
Let's say.
microbe will die. How microbe will die if they do not have external electron acceptor?
So who is they have to transfer this electron to somewhere, right? So after the for example, like I give you food, but I don't allow you to breathe oxygen, then you are gonna die, right? The same, it's as simple as this. But okay, so let's discuss, let's go through the slide, okay, and I'm going to explain the thermodynamic significance, and then we'll come back to your question and see whether it's clear or not, and we can have more discussion, okay. So we can actually, so we learned so far that actually we can have electroactive bacteria on our anode, okay, biofilm, and we can actually provide them with electron donor, and we know that waste and wastewater, they are very good electron donor, okay. You have a wide variety of organics, and we can actually get different types of value added product if we can manipulate the terminal electron acceptor. So the question is how we can actually get different types of value added product. So the first application of this technology was microbial fuel cell. So what is microbial fuel cell? You have anode biofilm, they oxidize the substrate or electron donor, release electron and transfer electron to the electrode surface. Electron gonna go to the cathode, and you have oxygen, and cathode oxygen reduction happen, and then you have an electron flow, which is electricity, right? So you can have electricity that you can use. So this is the basic application. Then researcher thought, okay, you know what, electricity, sometimes people consider, okay, this is cheap, right? We want to get more high value from our waste or wastewater. Then researcher thought, okay, what about producing hydrogen instead of electricity? Okay, so what if we kind of use the electron and convert that into hydrogen? So how we can produce hydrogen on cathode? So we can have proton, and we can have electron that is actually coming from anodic oxidation, and then we can have this half reaction. So proton and electron can be combined together, and you can have hydrogen production. But for this process, we need to supply energy from outside. And why is that? Because it's thermodynamically not possible. So typical applied voltage is about more than 0.25 volt, but you can see now you can say, okay, you know what, if I provide energy, why I don't just do water electrolysis, right? Why I should do microbial electrolysis, right? But if you compare with water electrolysis, you need at least 1.8 volt, right? More or less 1.8 volt, but here maybe within 0.25 volt you can get hydrogen production. And you have to also remember that at the same time you are treating your waste and wastewater, right? So we are actually getting dual benefit. So we are treating our wastewater, extracting some energy or electron, and at the same time you are providing some additional electron to get biohydrogen. But why we need to apply voltage there? Okay, so look into, if we use simple acetate, okay, simple acetate as electron donor in our process, what is the redox potential? Minus 0.2 volt. Sorry, minus 0.28 volt, right? So this is theoretical redox potential if you have oxidation of acetate, right? So if we provide microbes with acetate, minus 0.28 volt. But now if I want oxygen reduction to water, what is the redox potential for this reaction? It's 0.82 volt. But if you want to produce hydrogen, okay, if you want to produce hydrogen from proton, this redox potential is minus 0.41 volt. Okay, so now delta G we can calculate from number of electron transferred, okay, and Faraday's constant, right, F, and we can have redox potential for acceptor and redox potential for donor. But even without calculation, what you see? So electron acceptor minus donor. So if I simply write, so delta G minus NF E acceptor minus E donor. So minus NF. Okay, let's talk about MFC. MFC, what is acceptor? 0.82, right? 0.82. And what is donor? Minus 0.28. Here, this is acceptor. Okay, so now this is going to be positive or negative, this delta G value? This is going to be negative, right? But how about for hydrogen? For hydrogen it's going to be positive, right? So that means for hydrogen it's not thermodynamically feasible because its delta G going to be positive, right? So what we need to do? We need to, and how much the difference? So minus 0.28, okay. So this is going to be, let me write it, let's do it for hydrogen because I want to show you how we calculate how much energy that we need. So here acceptor is minus 0.41. What is minus 0.41? And this is 0.28. So what is the, so minus, so that means minus 0.41 plus 0.28. So what would be that value?
Minus 30.
Right?
Minus 30.
Minus, yeah, minus 0.013 volt. So that means that's the energy difference. So that energy has to come from somewhere. Then you can have that reaction happen. So if you like provide 0.13 volt from outside, it's gonna reach an equilibrium, right? Delta Z is gonna be 0, right? But you need more than that. So minimum you have to provide some voltage more than at least 0.13 to actually kind of start the reaction to happen. Okay, so this is actually the thermodynamic significance of yes, you can decide, okay, I'm going to put this electron acceptor there and I want this product, but you have to look into whether you need energy to, you need to provide energy from outside or not. Okay, so here we are seeing that we need minimum of 0.13 volt, but if you provide 0.13 volt, do you think you are going to see hydrogen production? No. Why? Because there are going to be some other energy losses in the system. So there are going to be other resistance in the system that going to create some barrier for electron flow, right? So there are going to be more energy losses, and those energy losses we call them ohmic or internal energy losses. Okay, so they can be contributed by different component in your system. So for example, we have membrane, right, between anode and cathode, and anode and cathode, they are far from each other. That distance can also contribute to some energy loss. At the same time, like solution, you have electrolyte, right? That solution also going to actually like create some barrier for charge transport, right? So those energy losses caused by those factors are called ohmic losses. So that means if we provide theoretical energy that is required for reaction to happen, okay, that's not going to be enough. We have to provide enough energy to also make sure that that energy input can also compensate for these kind of energy losses in the system. Okay. So any questions so far? Okay. So I think yesterday I was talking about like we can combine this process with also like anaerobic digestion process. So for example, like anaerobic digester, we can introduce electrode and we can apply some voltage, okay, to generate hydrogen, okay, and we can have you know, like a better process. So how it can be better? So you can see here in anode we have electroactive bacteria, so they can actually oxidize you know, like a compound like acetate, okay, and then on the cathode surface some methanogen, they can either use hydrogen or some methanogen yeah, they are hydrogen trophic, but some methanogen, they instead of using acetate or hydrogen, they can directly take electron, okay. Some methanogen, they can actually directly take electron, and it has been proved over last 10 or 15 years. So now yesterday we learned that actually there are two types of methanogen, right? So what are those two types? Acetoclastic and hydrogen trophic, okay. So there is third type of methanogen. Now we call them electrotroph. Why electrotroph? It came from, you know, heterotroph. What is heterotroph? They use organic carbon, and electrotroph means they directly use electron. So some methanogen, instead of hydrogen or acetate, they can directly utilize electron, okay. And you can see here, you can have this kind of reaction. So hydrogen can be produced from reduction of proton, and then that hydrogen can be used by hydrogen trophic methanogen. On the other hand, some methanogen, they can directly utilize electron from the electrode surface and produce methane, okay, and that has all.
I have a doubt. So yesterday you told us methanogen is not a good term, right?
Methanogen, no.
Because it's archaea.
Yeah, methanogenic archaea. I said we cannot call them bacteria. Okay. Although they have similar feature like bacteria, we call them methanogen or archaea.
So
okay.
methanogen is okay.
Yeah, methanogen is okay, but you cannot say methanogenic bacteria.
They are the type of bacteria, but
They are not type of bacteria.
they are actually not the bacteria. They are type of bacteria because
I guess.
No, they are type of microorganism, but they are not type of bacteria.
I
Okay.
guess it is just like bacteriophage.
No, yes, like as I was saying, like 30 or 40 years ago, researchers used to call them bacteria because they were almost similar, you know, like metabolic process, similar size and shape like bacteria. But later on, they found actually they are different. They are not bacteria.
but the archaea we only can find in extreme conditions.
Extreme, they can survive in extreme condition, yes, but they are not bacteria. Okay. So yesterday what I was telling that we cannot call them bacteria, we can call them methanogenic archaea or methanogen, that's fine. Okay. But here I am saying, because the thing is that every, you know, like in every 10 or 15 years researcher, they find like new feature of microbes, they try to give them new names, right? So for example, electrotroph, it's kind of like, you know, it became very popular term in over, I would say, last 10 years. Okay. So if some microbes, they can take electron directly, we can call them electrotroph. Okay. But you know, nonetheless, so what you have in if you have make AD, so in the conventional AD, what happened? We learned what happened. We have hydrolysis, right? You have fermentation, and then acetate is consumed by acetoclastic methanogen, and hydrogen and carbon dioxide used by hydrogen trophic methanogen, and you have methane production in your digester. But when you have make AD, what would happen? Those acetate gonna be mainly utilized by electroactive bacteria, and that is kind of like desired. Why? Because we know that the growth rate of acetoclastic methanogen is very low. Even compared to hydrogen trophic methanogen, their growth rate is very low. Okay. But if you compare EAB and acetoclastic methanogen, you can see, so for example, Ks value for EAB is 20 to 190 milligram per liter BOD, but for Ks value for acetoclastic methanogen, if you remember from our day 2 slide, from 180 to 450. So which one is kinetically efficient? Low Ks value, right? So if something has low Ks value, this is kinetically more efficient. So that means if someone, so it's like, let's say you can run faster, right? So you can beat me, you can outcompete me. So the same thing happen when you have EAB kind of growing in your electrode surface, they can outcompete acetoclastic methanogen for substrate.
So EAB is nothing but ETB, electrotrophic bacteria, right? EAB.
Electroactive bacteria, EAB.
Kind of whatever we have studied.
Yeah, I know that it's confusing. Like so, if so, EAB typically we use if something can transfer electron, okay, like extracellular using EET mechanism, we call them EEB. If something can take electron, okay, by extracellular electron transfer, then we can call them electrotroph, okay. So yeah, many terms in this area.
EAB a subclass of ETB.
No, that is different because EAB is on anode side and ETB on cathode side, right?
No.
ETB on cathode side, yes. ETB, I never use the term ETB. So electrotroph means they are taking electron, and electroactive means they are transferring electron. Yeah. So actually EAB you can actually have like perfect actually monolith pattern. We are going to actually discuss about that in later slide. But as you can see here, so what is for aerobic heterotroph? In day 2 actually we discussed that, right? So aerobic heterotroph in activated sludge process, their Ks value is from 12 to 60 milligram per liter BOD, right? So still those aerobic activated aerobic activated sludge process microbes, they might be kinetically more efficient, but compared to conventional anaerobic microorganisms like methanogen, that EAB, they are kinetically more efficient, okay. So still that's why researchers, they are thinking that these kind of microbes, okay, we can have anaerobic process for wastewater treatment, okay, and we can have possibly achieve low BOD concentration in the effluent, which may not be possible with regular anaerobic process. So another application could be microbial electrosynthesis, because these days there is lot of interest on like converting carbon dioxide, carbon capture, right? So converting carbon dioxide to some value added product, okay. So and we can also do that using this kind of process, but here your anode gonna be abiotic. So you can have anode, we can have water electrolysis reaction that can release electron, and electron can be transferred to the cathode surface, okay, where you have methanogen like same as MAC-ID system, but here it's kind of separated by membrane, okay. And if we provide biogas, those microbes, they can hydrogen trophic methanogen, they also need carbon dioxide, okay. Electrotroph means they can take electron, but they also need a carbon source, right, for their growth. So they can use CO2 as their carbon source, and you can have almost like you can get rid of all the carbon dioxide, and you can get close to renewable natural gas. And some of you attended yesterday's research seminar, and actually I was showing even from our experimental data, actually we can completely get rid of carbon dioxide from biogas using this kind of process. So another application could be we can actually produce some value added chemical on the cathode, okay, and we can use that chemical for different application in wastewater treatment. So for example, one compound is hydrogen peroxide, which is a very strong oxidant, right, that can also be actually used for disinfection, okay. So how we can actually produce hydrogen peroxide? So you can see here typical MFC, what we have oxygen and 4 electron and 4 proton, they are combined together and produce water, right, and this is 4 electron reaction. But if you can also have, if you can manipulate or you can have a better catalyst, you can also have 2 electron reaction. So for example, 1 mole of oxygen, 2 mole of electron and 2 mole of proton can combine together and produce hydrogen peroxide, okay, hydrogen peroxide. So and then that hydrogen peroxide can be utilized in situ for different water or wastewater application. But this topic is, there are a lot of challenges, and why it's challenging, because already the proof of concept is done. Okay, there have been actually a lot of paper on this topic, but the major challenge was actually most of the time initially you are gonna see lot of peroxide production, but over the time peroxide gonna be decomposed to water again. Okay, so when you have lot of electron over time, so you are gonna see they are gonna be, so first you are gonna have 2 electron reaction. Okay, but when you are gonna have more electron, you are gonna actually, that peroxide gonna be decomposed to water again. Okay, so then you may not, so the one technique could be, as soon as it's produced you kind of utilize them in situ. Okay, so you don't wait. If you wait, okay, you know what, you are going to extract the chemical, leave it, it's going to be decomposed to water. So that's why researchers thought, okay, still this could be a feasible approach, but you have to use that hydrogen peroxide in situ. Okay, so so that means your cathode chamber, okay, anode chamber can be a regular anode chamber where you are going to have EAB, they are going to use organic compound and you know, like transfer electron. But cathode, we can actually have we can actually feed our wastewater, okay, with different types of contaminants. So for example, peroxide, if we add iron salt, that is actually a strong oxidant, we call Fenton reagent. Anyone familiar with Fenton reagent, right? Yeah. So peroxide, it's a strong oxidant, but if you add iron, okay, we call it Fenton reagent, which is even better oxidant. Okay, so if we have, and that can actually, oxidant can generate hydroxyl radical. Okay, so and you can actually use that thing for advanced oxidation process, and I'm pretty sure some of you might be familiar with advanced oxidation process, right? So what we can do wastewater treatment. If we have very recalcitrant compounds, something cannot be degraded biologically, we can actually use Fenton reagent or hydroxyl radical to degrade them. So for wastewater treatment plant, the final step, what is the final step?
Chlorination.
Disinfection, right? So we can actually have even the final effluent, we can actually pump to the cathode chamber and we can have disinfection there. Okay, in situ disinfection. Sometimes that hydrogen peroxide, some researcher also demonstrated that, okay, you know what, like anaerobic digestion process, sometimes hydrolysis can be rate limiting. Okay, so we can use this kind of strong oxidant to hydrolyze the organic, particulate organic. Okay, so even we can use this method to hydrolysis of like sludge or any other feedstock that we use for anaerobic digestion process. Okay,
So kind of pre-treatment.
kind of pre-treatment, yes. Any questions so far? Okay, so let's move on. Another thing we can do is desalination. So every year we see new application, okay, in this topic. But why I'm laughing, I'm gonna you know, tell you later. But so how we can actually achieve desalination? Okay, so desalination we can have, instead of 2 chamber. So far what we have seen, like 2 chamber, right, anode and cathode. But for desalination, between anode and cathode chamber we can introduce a third chamber. Okay, and why third chamber? Actually third chamber can be used for desalination. Okay, so how we can achieve desalination? So we can put, so typically anode and cathode chamber, they are separated by membrane, right? But what we have, so let's say, you know, like seawater, what we have? We have sodium, we have chloride, right?
And bromide.
And bromide. So basically some cations, some anions, right? So what if we put an anion exchange membrane in one side of this chamber and another side we put cation exchange membrane? Okay, so what would happen when you are going to introduce saline water? That anion exchange membrane going to promote transport of anion from this chamber. And why? Because of concentration gradient. You don't have enough chloride in there, right? So because of concentration gradient, that chlorine gonna transfer from the middle chamber to the anode chamber. On the cathode side, you have cation exchange membrane. So sodium can actually transfer from this chamber to the cathode chamber to because of concentration gradient. Okay, so that means you can get rid of, if you kind of recirculate continuously over time, you are gonna see all chloride and your sodium kind of like transferred, they kind of disappear from the water, and now you have actually desalinated water.
So if you remove microbe from this system, it will be nothing but electrodialysis. It is a same
Yes.
system.
Yeah, so lot of feature actually adapted from other electrochemical processes. Okay, so saline water, and similar concept, actually a researcher, they demonstrated that we can actually get water softening. So for example, like in some cities, water can be really hard. Okay, water can be really hard, so you are gonna see your plumbing system can be damaged over time, and sometimes when you have hard water, you don't feel good after taking shower, right? Your skin is very rough, feel very rough, right? So that's why water softening is a very popular process. But most of the time, water softening process, it's kind of like done by using resin, right? Using resin, and you use salt water to actually regenerate the resin. So which is a common process. But actually researchers demonstrated that if we actually provide hard water in the middle chamber, we can get rid of the ions like that contribute to the water hardness, like calcium, magnesium and other minerals, and we can get soft water from the process. Okay. So another thing we can achieve is nutrient removal and recovery. Okay. So how we can do that? So let's say we can have source separated urine. Okay. So we can have source separated urine, and in our urine what we have? Nutrient. We have nitrogen, we have phosphorus, right? So we have nitrogen more or phosphorus more? Nitrogen more, right? So what we can do, we can feed urine in the anode chamber of the MAC reactor, and we can have a cation exchange membrane. So if we have cation exchange membrane, ammonium, it's cation, right? So because of concentration gradient, some cation like ammonium gonna transfer from anode to the cathode chamber. Okay, so then you can actually collect those ammonium from your cathode chamber, and that can be used as a liquid fertilizer. But now phosphate, okay, this is anion, right? So phosphate cannot actually move from anode to cathode. So in the effluent you are gonna have phosphate, okay, and you are going to have some remaining ammonia as well. Okay, so all ammonia are not going to be transferred, there are going to be some ammonia left. So if you actually collect the effluent and if you add some magnesium salt, you can actually get struvite, and I think some of you might be familiar with the term struvite. So struvite is actually magnesium ammonium phosphate, which is actually considered as a slow release fertilizer. Okay, so using this process we can also produce fertilizer. So we talked about denitrification, right? So what is denitrification? Nitrate to nitrite, right? So we can actually also achieve denitrification here. So how we can do that? Because some denitrifying bacteria, they can use hydrogen as electron donor, because denitrification process, you remember that we need to provide electron donor from outside, right? So because a nitrate is kind of like act as electron acceptor, okay, in anoxic process. So we can produce hydrogen, and if we put some denitrifying bacteria and if we provide nitrate, we can actually convert nitrate to nitrogen gas using denitrifying bacteria.
So could you explain this again?
Here?
Yeah.
So here it's gonna be MAC. So you are gonna provide power supply, okay, and you are gonna produce hydrogen, and you are gonna add some denitrifying bacteria, okay, they can form biofilm there. So you are gonna have in situ hydrogen production, okay, and in cathode feed it's gonna be maybe some water, wastewater coming from your aerobic tank, okay, or aeration tank, where actually you already converted ammonia to nitrate. Okay. So then you have denitrifying bacteria, you have nitrate from wastewater, you have hydrogen from the cell mag, okay, which is electron donor, and you have electron acceptor nitrate, and you can have denitrification. So basically, if I try to explain it more, so let's say you have electrode, okay, you have denitrifying bacteria. So from the reaction, electron is coming to the cathode, right? And you have proton, and you are actually converting that to hydrogen gas, which is an electron donor, hydrogen, right? And when you are actually feeding, let's say you have an aeration tank, you provide oxygen, you converted already all ammonium to nitrate, right? So if you pass this, let's say you have a cathode chamber, okay, you are actually feeding, you are recirculating between two tank, okay. So you are basically feeding nitrate there. So nitrate is electron acceptor there, and you have electron donor, and you have bacteria. So you have everything to get denitrification, right? So and then actually effluent can go back to your aeration tank for next process.
So in that effluent, what is there, which is going back to aeration tank?
Oh, it's then there is no nitrogen there. You completely remove them, right?
So clean one.
Yeah, clean water. Yeah.
And what happens?
It could be actually also used as a polishing step, right? Not necessarily you have to just. I was just showing an example, but it can be a polishing step.
And what happened to hydrogen here?
Oh, hydrogen is consumed in the process as electron donor. Yeah. Actually, there are some research on hydrogen, using hydrogen as electron donor. Actually, I think one group, even the lab I did PhD, actually they are also working on hydrogen as electron donor. But there are few groups actually working on that. So another application could be biosensor. Okay. So what is biosensor? Because those microbes, okay, so they kind of follow monod pattern. Okay. So if you increase the substrate concentration, the electrical response gonna increase. Okay. So if you actually increase the concentration, so you can actually get a perfect calibration curve. Okay. So let's say so what is monod pattern? So monod pattern means if, so what we learned, you have substrate concentration, let's say COD, right? And what is monod pattern? With increase in substrate concentration, growth gonna increase and reach maximum, right? So now this is growth, but here instead of growth we can consider electrical current, because those microbes, they provide like responses electrical current. Okay. So we can delete that, okay, and we can put current density. So if your COD or any contaminant level in within this range, okay, you can actually have a perfect calibration curve. Okay. So and you can actually use that concept as a biosensor. Okay. Sometimes we kind of use as a biosensor for readily oxidizable compound, okay, but sometimes you might have toxic compound. So if you have a clean baseline and let's say like suddenly your current decreased instead of increasing, so you can tell, okay, you have some toxic material in your wastewater. So this concept can be used for like to monitoring whether there is any toxic like compound is coming with your wastewater or not. So
I have a question here.
yeah.
Suppose in this kind of system, because bacteria is there, so they are also using energy to grow and maintain their cells. So by increasing the number of cells, don't you think the baseline will be changed? Or electrical response?
Electrical response में
So,
so to answer your question, so first you have to actually reach a baseline. Okay, so if you have a continuous flow, so baseline means your wastewater, okay, organic content is same, right? They are getting same feed. You have biofilm grow and some there are going to be some decay. So it's going to be more like stationary phase, right? But now if you have something changed, okay, you might see like current going to increase, or if you have toxic compound, current might decrease, okay,
So we have
and you may reach a new baseline.
So we have to look for the decrement.
Yeah.
Right here.
It depends.
Because our job is to detect here toxic compound.
If toxic compound, yes, if you are targeting any toxic compound, current gonna decrease. But if you are targeting any, let's say you wanna see whether BOD is increasing or not, whether, so let's say I put this kind of system in the effluent line where actually I am discharging my treated wastewater in river, right? Now if you see one day current is 5 milliamp, but next day you see your current is 10 milliamp. So you might think, okay, you know what, your treatment process may be not actually working properly, right?
Because
So
more COD.
more BOD is actually going there, right? So we can actually apply this concept, and actually some commercial product using this concept has already been actually available in market. Actually, even I met actually one company few months ago. They are actually trying to actually we are trying to collaborate and see, you know, if we can actually kind of make some bios. So from my research group, actually we try to develop a biosensor, okay, for wells and process water. So we are trying to see if we can actually apply this concept, because most of the time wells and mining site, they are in remote site, right? So there you may not have like, you know, large analytical equipment where you can take sample and do it on site. But if we can actually develop something that we can actually work in, you know, like apply in the field, maybe they don't need to actually take sample and send it to those sample to some analytical lab. So based on the electrical response, they can kind of tell, okay, whether, you know, there is some seepage of any contaminant in groundwater or something happen in the process, okay.
I guess we can connect to the IoT and we can also get the data online.
Yeah, data online as well, yeah. So yeah, that part, so that's kind of what we are trying to do. So some commercial company, they are expert on this kind of, you know, like, so we can do bench scale research and kind of, you know, develop the technique, but we don't, you know, kind of develop the product, final product, right? So
Scalability.
yeah, scalability can, yeah, sometimes we need to collaborate. So this is, before I jump into like process parameter and kinetics, any question? Yes.
Sir, what will be the maintenance of the biofilm? The number of microbes can be increased or decreased in such conditions.
If you have you know, like if you have, let's say, maintain them with same concentration of electron donor, okay, it should actually like it should not change, because they are going to be, it's going to be more like stationary phase. If you see microbial growth curve, it's going to be more like stationary phase. So growth going to be same, growth and decay going to be kind of same. So it should be stable, okay. But why I was laughing, okay, so you can see there are a lot of you know, promising result, okay. There are a lot of different things that you can achieve with this you know, area, but still there are a lot of challenges, okay. And one of the major challenges is how you scale up this kind of system. So when you you know, do some experiment in the lab, you work with small you know, like a small reactor, right? Small reactor, that means your ohmic loss is small. But when you make a large reactor, okay, you have a significant energy losses in the system. So now the question is how we can scale up, because I can do experiment in, let's say, 500 ml reactor, but for wastewater I need few 100 meter cube, right? So how I achieve that? So that's kind of like the main challenging part here, and still I would say we are kind of in learning phase, okay, fundamental learning phase. What happened when that topic became very popular, okay, in early 2000, okay, researcher thought, okay, you know, when there is some hot topic, everyone wanna do the same thing, okay. So what they do, okay, wastewater A, they try to see, okay, it can produce current or not. Wastewater B, you try that as a, so more like focusing on treatability study. And those treatability study actually doesn't help that much to, so I must say, okay, you know, I'm not criticizing any researcher, but I must say this is not a good approach if we want to scale up something. So we should focus on fundamental and try to see what are the challenges and then overcome one by one. Yes, product like biosensor, because biosensor can be a small reactor, right? What I did in the lab, the same thing can be in the field, same size, right? So it's not that challenging, and that's why the idea of biosensor has already been commercialized, okay? But if you are thinking about wastewater treatment for a community or something, then it could be challenging. We have to think about, okay, you know, how we can scale up this kind of idea, okay? Any other question? All right, so let's move on. You want to take 5 minutes break? Okay, let's take 5 minutes break and then we'll continue. This is, I have another 15 or 16 slides, okay? So let's take 5 minutes break.